So for today's lecture is on plant secondary metabolite and also plant defense and also plant defense. So as for a short introduction here, so plant secondary metabolite. So you are wondering why the names is plant secondary, not plant primary. So plant secondary metabolites can be differs can be distinguished between these secondary and also primary because this secondary usually they are the overspill from these primary metabolites. So if you watch the video I uploaded in the e-learning, so the one that the compounds ataupun the metabolites, the one that plant produce, the compound that plant produce can be divided into two kinds of group. This is a big group. Actually, if you try to classify these plants compounds group ataupun plant metabolites group, there are a lot of category. So you have to know there is if you look at a large the initial of this the large perspective of these compounds or metabolites, we usually look at these two things. Either they are primary, these primary compounds usually they are directly contribute to the plant growth and the development. So those compounds or those metabolites, we call them as primary. So example of these primary metabolites are carbohydrate, protein, and also lipids. So I last time you learned this thing since the first year you learn also, but now we are focusing on the plant. So okay, we leave behind this primary metabolite. So now we are looking to this secondary metabolite. So secondary metabolites are the other compounds that does not contribute primarily. That's why they call they are not contribute primarily to the plant growth and development. So last time we learned about plant nutrient, and also if you still remember, I teach you about calcium, nitrogen, phosphorus, and so on. Those are primary metabolites needed by the plant. So now we are looking by the compounds ataupun the metabolites produced by the plant secondarily. So later on we are going to see why these plants are producing these secondary metabolites. So what else before I'm okay, a little bit I have to say history on these secondary metabolites. So the term secondary metabolites was first introduced by one German biochemist named Albrecht Albrecht Kossel. So in 1910. So later on, if you have time, you can Google and so on. On 1910, 1910. So this biochemist, he won a Nobel Prize because he is the first scientist or biochemist who introduced this term of secondary metabolite. Usually, maybe long ago, they only call these things is compounds. Carbohydrates is compounds, proteins is compounds, and also this secondary metabolites is compound. But later on, this biochemist, he grouped these secondary metabolites into one group called as secondary metabolites. So because of these compounds, they are less essential and also they are of this secondary relevance. So what does this biochemist discover? So I think you guys might know about this upper compound. So he discovered that nucleic acid. So I hope you remember what is nucleic acid in your plant. So nucleic acid is one of the organelles where all the genetic materials inside. You have your plant cell, so you have your cell wall, and then you have so many organelles there, and then one of it is the nucleic acid. So this nucleic acid contain your genetic materials ataupun not your lah, the plants lah, because we are looking at the plants. So inside this nucleic acid, you have a chromosome and so on. So if you this Kossel, Albert Kossel this German biochemist, he looked inside this chromosome and he found out that this nucleic acid is composed of 5 nitrogen bases. If you still remember what is the nitrogen bases in nucleic acid, so we have 5, which are adenine, cytosine, guanine, thymine, and also uracil. So Please read further. We actually in DNA we have 4, RNA we have 4. So 4 of this nitrogenous base. So please search more. What does the nitrogenous base present in the DNA? What is the nitrogenous base present in the RNA? So he's the one who discovered all these 5. If you are wondering who discovered this one, this base is found by this cell. So since these compounds they are contribute into the less essential of this plant compound, so we call this as a secondary metabolite. So what else to make you understand about this secondary metabolite? Oh, if you still remember in the previous laboratory practical, we do TLC in our lab. You still remember? We going to we do the TLC to see the pigmentation of chlorophyll A, chlorophyll B, xanthophylls, and also apa yang last kali hujung tu, beta carotene. Okay, those pigment you quantify last time during TLC experiment, those are secondary metabolites. So secondary metabolite, you can in some biochemists they call them as phytochemicals. And also in previous lecture also I mentioned about this terpenoid, essential oil, and so on. Essential oil is one of the secondary metabolites. Okay. So this plant secondary metabolite, it is a very interesting compounds to be studied and investigated. So until now, so if we are looking at the research perspective, so until now, whenever they found a new plants, for example, kan, if maybe some of you are very interested in plant and also love to go to hiking, and you found out that, oh, I saw one types of orchid and also quite rare orchid. So usually apart than looking at their morphological characteristic, scientists also are very interest on looking for their biochemical compound, which is their secondary metabolites. Who knows, we can use their secondary metabolites for the many other purposes, such as for the medicinal purposes, for the therapeutic purposes, and also, for example, essential oil also, we are using them for aromatherapy purposes. Okay. So hope you guys have a view on this secondary metabolite. Okay. So what is the difference between primary and secondary metabolites? I mentioned you just now. Hopefully you can differentiate between these two. All right. So these are the pathway of this upper production of secondary metabolites. Starting from this, we are looking at this the 3 main primary metabolites. So how do how do plants produce the proteins, the lipids, and also the cellulose, the form of the carbohydrate and the protein. So as we can see here, the upper secondary metabolites, they are the overspill of this primary metabolites. So here, okay, so you can see here how does this secondary metabolites are produced. For example, this one I don't want to go in depth because we have another pathway of these secondary metabolites, how they produce from the photosynthesis until each class of these secondary metabolites are produced. Okay. So what are the functions of secondary metabolites? So most of these compounds, they are produced in response to this biotic and also abiotic factor. So please search what is biotic and what is abiotic factor. So usually because plants, you see plants, they cannot move anyway. They are, just imagine you yourself, your feet are buried in the ground and also you cannot move anywhere. So sometimes this plant they have to produce certain compounds in order to respond to the environment. Okay, the biotic factor I mentioned just now is the example of the biotic factor are the attack from the pests and also diseases. So yes, they protect the primary metabolism by deterring the herbivores, reduce the tissue loss, and also avoid infection by microbial pathogen. Okay, other second function of these secondary metabolites also they attract pollinators and seed dispersing animals. So if you ask me how these compounds attract the pollinators and seed dispersing animal, the answer is easy. The color from the flower, if you see some flower tend to be purple in color and so on, some are red, some are yellow and so on. Those pigments contribute to the color of the flowers or color of the even the whole body of the plants. Those are from the secondary metabolite groups. So I give you one example. So if you see a purple flower, so we have one, I think one flower that are quite famous just now, not just now, recently lah. This flower has been used by those who are from, I think some of you who's from Terengganu and also Kelantan. You use traditionally we use bunga telang. So I know you know bunga telang. It is a purple color flower which is used by some people to for a certain people who cook nasi kerabu. So because of the color, we human being use this secondary metabolite properties to color our rice and also to make it a bit attractive. But in the perspective of the plants, they use this colorful flower to attract these pollinators and also seed dispersing animal. So the color of violet or purple color of the purple to violet color of flower usually is contributed by a compound. So this compound is categorized under secondary metabolite, which is we call them as anthocyanin. You can even search more about this. Very interesting lah about this. And also for those color, for example, color orange to red color. So usually we relate this compound with we relate this color into to a pigment or a compound we call as betanin, Betani. Later on you can search more. So a lot of interesting compounds in the plants. Okay. And also they also act as an agent of plant-plant competition. So for this agent of plant-plant competition, we look at for example, I mentioned this just now, the aromatic plant, the essential of plant. Those plants are producing odor, attract more pollinators and also seeds. So sometimes in order for them to compete among themselves, so some plant need to show some extra character so that they win that place, something like that lah, right? Okay, so I hope you can imagine all these secondary metabolite. Actually there is a lot more if you are looking at the purposes and also the uses, the purposes, uses, and also the properties itself. Okay. Some secondary metabolite nowadays, the research trend is they are looking for the secondary metabolite that can cure cancer. So it is quite research yang quite macam kita kata not easy to do, but researcher are keen to search for a new medicinal plants and also they are testing those extract of the plant into some of the cell cancer and also later on they're going to apply on human. So there is a lot of research going on these plant secondary metabolites. So the other roles of these secondary metabolites is to display antimicrobial activity, protection against herbivory and also pathogens. This is quite interesting things. You can even consider to do it for your final year project. So last time I have my student who extract the essential oil. You can even extract your essential oil by yourself. So for example, you have a mint plant at home or any other plant that usually if you want to extract the essential oil, you have to choose the plant which have quite a strong odor. For example, last time I extract from the rhizome of the hali apa Zingiber officinae, ginger, ginger and also curcumin. So you can extract this essential oil and then you can test on the certain antimicrobial. That's why some researcher come out with this antiseptic natural antiseptic, maybe they extract from mint, something like that. But before they come up with this product, they have to test first on the bacteria. You have to culture the bacteria on the plate, and then you have to culture the bacteria, and then you need to put this your plant extract or your essential oil on maybe on the round paper. Usually I use filter paper and then just paste on your culture plates. So you can see the inhibitory response of this bacteria on this compound on your plant extract. Number 5 is for attractants, for smell, color, and taste, for pollinator seed and dispersing, and number 6 agents for plant competition. And also, there is also a plant response to the herbivore. So plant damage by insects can release volatile chemicals. So volatile chemicals is essential oil I mentioned just now. So essential oil are the volatile oils. So you can check also what is the difference between this volatile oil and also the normal oil yang you use dekat kat dapur lah. Ada perbezaan dia. Tak apalah saya bagi tahu sekarang lah. So for this essential oil, this volatile oil, they are aromatic and also kalau you remember you learn in your class before, kalau yang oil yang biasa yang from lipids ya, those yang betul-betul vegetable oils ke ataupun apa lagi minyak masak you all, it can saya lupa lah yang ada kertas, you punya kertas tu, it can produce a spot in your paper. So dia akan menyebabkan you punya paper bukan berlubang lah, dia akan jadi macam nanti please search about it. Whereas for these volatile oils, they are different because if you put on the paper, they will not produce this that kind of spot lah, that kind of oily spots. Okay. So this volatile chemical can also function as an early warning system for nearby plants of the same species. Also, methyl jasmonic acid can activate the expression genes involved in plant defense. So my friend did this jasmonic acid. So she used this jasmonic acid. Jasmonic acid is not a big compound. They are not big compound. They are not the primary metabolite. They are considered as secondary because whenever the plant was attacked by insect or any pest, they will trigger the production of jasmonic acid to be more. There is a certain pathway of it. Okay. So the research that she done is we can buy, actually this jasmonic acid you can buy, very expensive. So what she do is she compare between she dilute this jasmonic acid into a certain concentration and then she spray on the plant. So she compared the one that sprayed with jasmonic acid and the one not sprayed with jasmonic acid. The one that sprayed with jasmonic acid, they produce more fruit and they are tolerant to the disease. Alright, herbivore damage can elicit a signaling pathway. This is the induced defense. This is how the pathway for this jasmonic acid happen. There is a lot of pathway. So the one that we are showing here is for jasmonic acid pathway. How this okay, for example, herbivory meaning that grasshopper, maybe they bite your plants and then the plant will release a hormone called as histamine hormone, and then they will bind to the receptor of the plasma membrane, and then so the lipase will, if you still remember last week we learned lipase, lipase is one of the hormone triggered enzyme. So the histamine hormone, will trigger this lipase to break down the membrane lipids into fatty acid and also glycerol. So fatty acid, under fatty acid is linolenic acid. So this linolenic acid will then be synthesized into jasmonic acid biosynthesis to produce a jasmonic acid. So this jasmonic acid will be produced more in the plant to respond to this herbivory attack. Alright. This is one of the pathway jasmonic. We have a lot more compounds if you want to focus to other compound lah. Yang agak famous sekarang ni jasmonic lah. Alright. Okay, jasmonic acid, the level of jasmonic acid. Okay, just now I mentioned kalau lepas that pathway, and then the level of the jasmonic in the plant cell will rise up, and then this hormone will trigger many types of plant defenses including terpenes and alkaloid. So jasmonic acid pun dia ada other pathway, dia akan produce terpene and also alkaloids. The action of jasmonic acid induce the transcription of many genes involved in plant defense. So I guess ada juga recently ini lebih pada gene expression lah. So other lecturer also are doing this, quite interesting also. So jasmonic acid turns on genes for proteinase inhibitors. So this is apa? Ni pun sama juga macam yang tadi tu. So saya tak letakkan dia punya flow. Hopefully you guys can remember the flow. Who knows, I might come out this in your test ke apa ke, macam menarik lah. Kalau pathway ni memang sangat menarik, right? Oh, I think that's it for today.